4, 3, 2, 1.
First, we want to take a look at the plasma membrane, known as the cell membrane, which is known to be selectively permeable, representing a fluid mosaic model. It has 2 layers of phospholipids that control what enters and leaves the cell, meaning that it has the ability to keep out the bad stuff and let in the good stuff. We'll go a little more into detail about the cell membrane and how it's composed of a lipid bilayer and what that lipid bilayer really is. The cell membrane is composed mainly of a lipid bilayer. This is what helps it become selectively permeable, dynamic, flexible, easily repairable, and it decides which and what things to let out of the cell and what to let into the cell. So these things that you see right here, these circles, as well as their tails right here, that is known as a phospholipid. So the great things about phospholipids are that their fatty acid tails are not bonded, which allows for lateral movement in the cell membrane, allowing it to be flexible and represent the fluid mosaic model. They have a hydrophobic interior, so these tails are hydrophobic, meaning that they do not like water, and it prevents passages of polar substances, aka water-soluble substances, through the membrane. In sharp contrast of the hydrophilic tails, they have hydrophobic ends, which means that these proteins are embedded in the membrane, providing passageways for polar molecules to pass through. So if you're wondering what these things right here are, well, those are the protein membranes. So just going further into detail, you have your integral proteins, which work as channels and carriers to move substances across the membrane, making it easy to go in and out of the cell. And then you have your peripheral proteins, which act as receptors that detect and receive chemical messages from the cell. They also serve as identity markers that support carbohydrates, which identifies the cell by type. In addition, there's also cilia, which is located outside of the cell membrane, and they're short hair-like structures on the free surface of some cells. Going deeper into the cell and, really the pit of where everything is controlled, we take a look at the nucleus, which is right here. And the nucleus's purpose is to hold genetic material because it's the control center of the cell. It is the brain of the cell. It tells the cell what to do. Going even further into that, there's a nucleolus. And this nucleolus is a structure in the nucleus that helps assemble ribosomes. The thing to know about ribosomes is that they don't have a cell membrane and that they are assembled by 2 organic compounds, proteins and RNA. What really happens in the creation of ribosomes is that the cell nucleus compounds are packaged into ribosomes, so that's how they're made. And then they are transported into the cytosol of the cell, and they attach to the rough endoplasmic reticulum. Now, this relates to the endoplasmic reticulum because mainly the rough endoplasmic reticulum, which is ribosome bound, usually serves as a way to transport these proteins to go to the Golgi apparatus so they can be synthesized and processed and packaged outside the cell through these nuclear pores, which are the ugly circles that I drew. Okay, there's also a smooth endoplasmic reticulum, which is right here, and that is not ribosome bound. In fact, there are no ribosomes on that. So it does get a little confusing because it kind of looks like the Golgi apparatus, but when you're looking at it on a diagram and you don't really know how to label which is which, well, I usually just go with the fact that the smooth endoplasmic reticulum is always closer to the rough endoplasmic reticulum because I don't think your teachers really want to screw you up if they're making you identify what part of the cell is what. Nice way to look at the Golgi apparatus is that it's like a stack of pancakes, or at least that's what I like to think about it as. The important thing to know about the endoplasmic reticulum, I spelled it wrong there, so do not take my spelling into account, but is that there are 2 types. There's a rough endoplasmic reticulum, which is covered in ribosomes, and there's a smooth endoplasmic reticulum, which is free from ribosomes. So when you look at it on a drawing, the endoplasmic reticulum has a rough kind of sandpaper surface, and the smooth endoplasmic reticulum does not have any bumps on it. It's just smooth. So the function of the smooth endoplasmic reticulum is to regulate calcium levels and synthesize steroids and regulate toxic levels as well. Moving on, there's also the rough endoplasmic reticulum. Now this is a little bit different because its function is to prepare proteins for exports. So this rough endoplasmic reticulum serves as highways where molecules move from one part of the cell to another. The rough endoplasmic reticulum mostly works on transportation. Where does it transport the ribosomes? Right to the Golgi apparatus. So the main function of the Golgi apparatus is that it processes and packages substance produced in the cell. So it takes those proteins and it processes and packages them so they are ready to be exported out of the cell through the nuclear pores. Then we have the mitochondria. Its function is to transfer energy from organic compounds to ATP. Fun fact is that a cell with lots of mitochondria means it has a high energy requirement. So organisms that need more energy tend to have more mitochondria. So looking at the structure of the mitochondria specifically, it is surrounded by two membranes, as you see the first one, which is called the outer membrane. Outer membrane. And then right next to that is the inner membrane. If you go a little further into the cell, you can see the cristae, which is pretty much known as the long folds that provide space for chemical reactions. So all those compounds being transferred into ATP and transferring energy, that all happens in the cristae. Lysosomes, which you don't see here, are actually located around and near the Golgi apparatus, and those function as bags of digestive enzymes in the cell, which are actually known as suicide sacs.